So in this set of lectures, now we are going to go from where we stopped last time when we talked about the vesicles being released, opening of the vesicles, release of the neurotransmitter, and we talk how it can be uptaken by glia, going back to the neurons, and some of it binds to the receptors. So now we're going to talk about the receptors the neurotransmitters can bind. So in this first lecture, we are just going to introduce what metabotropic or G protein coupled receptors are and learn about the basic organization of this signaling. So what you have talked with Alfonso in the last couple of lectures was that neurotransmitter that's released can it bind ionotropic receptors, and hopefully by now you're the expert in ionotropic receptors, or it can bind metabotropic receptors. So what's the difference between ionotropic and metabotropic receptor? It's in the effect that neurotransmitter has. Neurotransmitter, when it binds to ionotropic receptor, it's going to lead to the opening of the channels. Tropic means movement in Greek, so ionotropic receptors binding of neurotransmitter is going to lead to the movement of ions inside of the cells. But in the case of metabotropic receptor, binding of neurotransmitter is to the receptor will lead to the changes in accompanying protein. In this case, these are G proteins, which will then lead to the activation of different effectors. So ionotropic receptors, again, you had several lectures on this, and we will just go very briefly over it. What is important for you to remember for this lecture is that this is a fast transmission. So the moment neurotransmitter binds, the channel is open and ions can move. They can flow over across the membrane. So it's around, the effects are time scale milliseconds. So it's very fast. Another important thing to know is the receptors and the ion channel are single molecule, and that's why it's fast. And these are different neurotransmitters and their receptors. So acetylcholine is going to bind nicotinic acetylcholine receptors, and you know that there are muscle and neuronal varieties. Glutamate is going to bind 3 different types, AMPA, kainate, and NMDA receptors. GABA has 2 different types, A and B. Glycine, serotonin, 5-HT3 receptor is ionotropic receptor, and then purinergic receptors, which are of P2X variety. On the other hand, we have metabotropic receptors. So they are either called metabotropic, or more recently they're called G protein coupled receptors. So in this case, again, when neurotransmitter binds, it's going to elicit the conformational change in this receptor that is then going to activate G proteins that are associated with them. These G proteins that are activated upon neurotransmitter binding to the receptor will then separate into several subunits, alpha and beta gamma, which are then going to affect different effector proteins, or they can directly act on the ion channels and lead to their opening or closing. In either case, this involves the sequence of several events, so this takes time, and this is why this is working, the metabotropic receptors are working on much slower time scales. So it takes hundreds of milliseconds to minutes to activate it, but also, as you will see soon, the effect of activation of metabotropic receptors lasts much longer. It can be visible much longer. What are the which are we talking about? So pretty much all neurotransmitters have metabotropic receptors to which they bind. So for acetylcholine, metabotropic receptors are muscarinic acetylcholine receptors. For glutamate, we have their names are metabotropic glutamate receptors. There are 8 different subunits. GABA B receptors are metabotropic. Dopamine, all of the dopamine receptors are going to be G protein coupled receptors. Serotonin are 1 to 7, so 3 is the only one that is ionotropic. Lots of different purinergic ITP and adenosine receptor A and P type are going to be metabotropic receptor. Histamine, norepinephrine, epinephrine. Also receptor for opioids, endorphins and enkephalines and other opioids are metabotropic receptors, as well for endocannabinoids and prostaglandins. Now here are some examples that will show you also the signaling is organized into several steps. So let's start with the norepinephrine. So norepinephrine binding to beta-adrenergic receptors, G protein coupled receptor, is going to lead to activation of GS, or these are stimulating G proteins. That means that they stimulate or activate adenyl cyclase, which is an enzyme that produces CMP. So cyclic AMP levels will increase in cells when norepinephrine binds to beta-adrenergic receptors through the activity of GS. And that increase in CMP is then going to work to activate later effectors, in this case shown example protein kinase A, which will increase protein phosphorylation of its targets in a cell. So you can see that they have several steps starting from neurotransmitter binding to receptors, activation of G protein that then exerts its effect of effector protein, making second messengers that then can activate later effectors and later targets. We have two more examples here. One is binding of glutamate receptor to mGluR receptor, glutamate to mGluR receptors, which in this case are connected with GQ proteins. GQ proteins, as we will see in the following lessons, are going to lead to activation of another effector protein, phospholipase C. Phospholipase C is going to catalyze the hydrolysis of phosphonazidol biphosphate into deacylglycerol and inositol 3-phosphate, which can then lead to downstream effectors such as protein kinase C activation and calcium release from the endoplasmatic reticulum. Both of these can increase protein phosphorylation and then also activate many different calcium binding proteins. And the third example that we have is binding of dopamine to dopamine 2 receptor. So this is type 2, which are associated with inhibitory G proteins or GIs. GIs are called inhibitory G proteins because they lead to the inhibition of adenyl cyclase, which is their effector protein. Inhibition of adenyl cyclase leads to a decrease in cyclic AMP levels, and this decreased cyclic AMP levels will decrease the amount of protein kinase A and therefore decrease protein phosphorylation. One of the key features that you should know that this stepwise activation allows is amplification. So this is one of the key features of the G protein coupled signaling. How does it work? So if we go from the binding of one neurotransmitter to the receptor, it can lead to the activation of many G proteins. So one activated receptor can bind to the activation of many G proteins. So that's the first step of amplification. These G proteins are going to lead, there is no amplification in this step, but they are going to activate as many adenyl cyclase enzymes, which will then again in the next step of amplification be able to produce many cyclic AMP molecules. Cyclic AMP is going to activate many protein kinases, so there is no amplification in this step, but then protein kinases, there is a huge amplification in this step. They can phosphorylate many different target proteins. So we have a tremendous amplification from neurotransmitter binding to one receptor to the amount of changed target proteins at the end. So action potential come to presynaptic neuron is going to lead to leakage of calcium inside, and when we have more agonists there, that is going to lead to G alpha I, so inhibitory alpha, which then beta gamma from this inhibitory alpha, when it separates, is going to lead to the closure of calcium. So therefore, the amount of neurotransmitter release is going to be much decreased. This same more agonist can bind to the more mu opioid receptor on the postsynaptic membrane. In this case, beta gamma can go and bind Kirk channels, as we have talked, and then it's going to lead to their opening. Potassium is going to leak out of the cells, and this is going to lead to hyperpolarization. So not only you can change the amount of transmitter release from the presynaptic receptor through activation of G protein coupled mu opioid receptors, but you can also hyperpolarize the postsynaptic neurons and then change its response to the amount of neurotransmitter release. In addition to directly affecting the channels or production of secondary messengers, G protein coupled signaling can elicit prolonged changes through transcriptional regulation. So as I told you, they activate much slower than ionotropic receptors, but their effects can last much longer. And this is one of the ways that their effect can last for many hours or days. So in this case, we have activation of protein kinase A that we have talked through. So we have binding of neurotransmitter. This is GS protein, so this is stimulatory, which leads to the stimulation or activation of adenyl cyclase, CMP production, and protein kinase A activation that we talked already. So this activated protein kinase A can phosphorylate Crab protein, which is transcriptional factor, which is activated when it's phosphorylated. It can go into the nucleus then and activate transcription. In this example, it can lead to the activation of messenger RNA that will eventually code for ion channels. So you can see that in this way, G protein coupled signaling can lead to the insertion of ion channels or ionotropic that will then change the way the postsynaptic neurons will respond to the neurotransmitter release. And finally, what I want to leave you for this first part is that neurotransmitters, as you can see, quite a few of them, such as glutamate, GABA, serotonin, they can activate both ionotropic and metabotropic receptor at the same time. So release presynaptic, they can on postsynaptic cell will bind to both of these different kind of receptor, both ionotropic and G protein coupled receptors. And response that's elicited by neurotransmitter will be determined by this complement of receptor that's expressed in postsynaptic cells, and also the associative factors that are going to be expressed in these cells, whether it's going to have which enzyme and which channel is going to have to be affected by these activation of these receptors.